So hello everybody, today I am going to discuss about the different methods of DNA sequencing. So let us have a brief introduction of this technique. There are different techniques of DNA sequencing. Let us have a brief introduction of DNA sequencing. Suppose we have a DNA here and we don't know the nucleotide sequences of this DNA molecule. These nucleotide sequences can be A, T, C, C, G, T, A and so on, or it can be T, C, A, G, T, C. So what exactly is the DNA sequencing of this DNA, the sequence of nucleotide of this DNA, we don't know. So we have to analyze or determine the nucleotide sequence of this DNA, and this is what we call as DNA sequencing. So DNA sequencing is determining the nucleotide sequence of a given DNA molecule, of a target DNA molecule. All right. So there are many methods which are being involved for this DNA sequencing, but here I am again limiting my lecture to only two methods, but in this lecture I will be taking only one method. So the first method which I will be discussing here will be Sanger method. Sanger method of DNA sequencing. This I will be using the first time. Sanger method of DNA sequencing. It was developed by Frederick, also known as Fred Sanger, and his colleagues in the year 1977. This method is also called as chain termination method, or it is also known as deoxy sequencing. All right. So let us take the ingredients of the Sanger method. What are the different components which we use? First of all, we need a target DNA which need to be sequenced. We should have the target DNA. Then we need a primer, which is a small stretch of DNA here, which is a small stretch of DNA from which the enzyme DNA polymerase can start the formation of new DNA strand. We need a DNA polymerase enzyme, and we need normal nucleotides, then we also need a special type of nucleotide which is called as deoxy nucleotide. All right. So let us have a difference between these two, normal nucleotide and a dideoxynucleotide. We have to see the difference between the two. So let me take this. So all right, let me insert a photo here. All right, so I am going to insert this picture. So if we have a study of this diagram here, then this is a normal nucleotide below, and this one is the dideoxynucleotide. Now, what is the difference between these two? In this normal nucleotide, this is carbon number 1, this is carbon number 2. So, on carbon number 2, oxygen is removed here. OH has to be there, so O is removed here. That's why we are calling it as D. D means removal oxy, and this is nucleotide, nitrogenous base, sugar molecule, and this is the phosphate. So, nitrogenous base and the sugar molecule, and the third is the phosphate. All these three, when combined together, they form the nucleotide. So, deoxynucleotide, but in this case, one molecule of oxygen is out from here, carbon number 2, and also from the carbon number 3 again, another molecule of oxygen is out. So, one oxygen is removed from this side and one oxygen is removed from this side. That is why we call it as dideoxynucleotide. Now, this type of nucleotide is abnormal nucleotide. It cannot combine with any further phosphate, or we can say it cannot combine with any further nitrogenous base. So, if we insert this dideoxynucleotide, then the chain is terminated. The formation of new DNA is terminated. So, this is the dideoxynucleotide. So, rest all the components are the normal components up to 4, 1, 2, 3, 4, which are required for DNA synthesis, DNA replication. All right. So, chain termination method. Let us see the steps. What are the different? How can we organize this Sanger method? Steps of Sanger sequencing. Now, we have to see the steps of Sanger sequencing. All right. So, let us let me insert one more diagram here. All right, this is. So, if we look here, it's gone up. All right, so I hope this diagram is visible to all of you. In the step 1, we have to take a target DNA. So, this one is the target DNA. Let me take some other color. Okay, this is the target DNA that need to be sequenced. So, first of all, we heat the target DNA and we denature it. Denature means we have to remove both the strands. We have to separate both the strands. So, we have to heat it at high temperature above 80 degrees centigrade or 90. Both the strands of the DNA get natured, separated, and they are denatured. Then in second step, we have to take a small segment which need to be sequenced. If we are supposed to sequence this one segment, then we have to take the one segment from here, and we have to make multiple copies of these segments. All right. So, then multiple copies means if we have taken the red one, then 1, 2, 3, 4, many copies of this. We can do it by PCR also. So, multiple copies of the target single stranded segment we have taken. Then we attach a small primer. Primer is a small DNA segment here in this case, and then we try to extend the DNA strands by adding the new nucleotides at the other end. And when we extend it, we supply the DNA polymerase 3, we supply the normal nucleotides, but we also supply the abnormal nucleotide at a very low ratio. Say for example, if it is here, it is T, and we give abnormal dideoxynucleotide A, so the chain will be terminated here only. All right. So we have to add 4 polymerase solutions here, 1, 2, 3, 4. In these solutions, we have added different types of dideoxynucleotides here in the very low concentration. Then we have to grow the chains. As I told you, here it is, the chain is grown. We have to grow the chains, and then the chain is terminated at those points where the dideoxynucleotide comes. And then these fragments are taken separately. And say for example, if we cleave this DNA with dideoxynucleotide adenine, then there are 3 segments. Say for example, one of the fragment is cleaved at 2 places, so there will be 3 fragments which are separated through gel electrophoresis. And by counting from smallest to the largest one, this is the minus terminal here, this will be the plus terminal here of the electrophoresis. So we can calculate the first strand of the DNA. Now, these steps are just the outline steps of this Sanger method of DNA sequencing. I am going to take this DNA sequencing in detail, but the steps are: first we have to denature the DNA, we have to make the multiple copies of the single strand, we have to attach a primer, we have to all these 4 are taken in different test tubes with the primer. We have, in these test tubes, all the 4 test tubes, we have to add nucleotides, normal nucleotides, we have to add DNA polymerase 3, and in all the 4 nucleotides we have to add dideoxynucleotide in very low concentration, different dideoxynucleotide. So the chain is terminated at the formation of new DNA is terminated at the dideoxynucleotide which has been added here in 4 different test tubes. 4 different deoxynucleotides are taken, and then there will be different fragments formed by the chain termination. These fragments are separated, and then from smallest segment to the largest segments, we just keep on giving names. Say for example, the smallest segment of T, just below the T, move the farthest, so it is written as T. Then we say this is when this one comes. This one is C. Then C will be there. So this will be the nucleotide sequence of the first strand. Then we can make the second strand. All right. So let us take another diagram to explain the Sanger sequencing in further more details. All right. So okay, if you take this diagram, if you look at this diagram, all right, I hope now it is clear. So what happens here in this diagram is something like this. All right. So this is the target DNA which has been taken here. This is the target DNA, single strand. Now this target DNA has a template over here. So we take the template on this target DNA, any one of them, any one of it. And this same sequence is transferred in 4 different test tubes: test tube 1, test tube 2, test tube 3, and test tube 4. This same nucleotide sequence, but we don't know the nucleotide sequence, but it is written here for the sake of convenience. So if we, in the first test tube, if we take dideoxy ATP, that means where there will be A, the chain will terminate there. Then starting from the first one, in front of A there will be T, C, but the third one is A. The 3 nucleotides like here, TCA. Okay, so it will be cleaved from here. Then again, wherever TT comes, in front of them A will come, and when the chain will get terminated there only, यहाँ पर भी terminate हो जाएगी. So 4 nucleotide sequence will be formed if we cleave this DNA with dideoxynucleotide, because at dideoxy the chain is terminated. Say for example, in the second test tube we take dideoxy GTP, and then there are only 2 fragments formed, dideoxy CTP and dideoxy TTP. There will be different fragments. These fragments are then separated through gel electrophoresis, and from lower to the upper, that means this is the minus side, minus terminal, this is the plus terminal of the electrophoresis. We just have to label from below the smallest particle to the largest particle. These bands are been labeled. Say for example, the farthest band is the T, and here it is written T. Then this is C, TC, the first one T as it is, and C. So like this we will have this sequence. The last one is the sequence, and this sequence is written here, and it is the first strand sequence of the DNA which we need to sequence. So that means steps of the Sanger methods are like this, and this is the detail of the Sanger sequencing. I hope you have understood DNA sequencing, finding out the nucleotide sequence. Thank you very much.